Hey everyone, welcome to the PowerPoint video for Module 9.3. We're going to be talking about some of the special senses here in much greater detail than we did in 9.1. There is a lot to cover in this PowerPoint, so this video is going to be broken up into two sections. In the first part, we'll cover olfaction, gustation, and vision, or at least a part of vision, and in the second part of the video, we'll cover visual physiology, equilibrium, and hearing. So let's go ahead and get started. So our knowledge of the world around us is limited to those characteristics that stimulate our sensory receptors. We may not realize it, but our picture of the environment is incomplete. Colors we cannot see guide insects to flowers, sounds we can't hear, and smells we can't detect give dolphins, dogs, and cats key information about their surroundings. What we do perceive varies considerably within the state of our nervous system. For example, during sympathetic activation, you experience a heightened awareness of sensory information. You hear sounds that normally you wouldn't notice. Yet, when concentrating on a difficult problem, you may be unaware of fairly loud noises. Finally, our perception of any stimulus reflects activity in the cerebral cortex. But that activity can be inappropriate. For cases of phantom limb pain, for example, a person feels pain in a missing limb, like we talked about in the last PowerPoint. During an epileptic seizure, a person may experience sights, sounds, or smells that have no physical basis. Here, we'll look at the 5 special senses, like I talked about, which are olfaction, or smell, gustation, or taste, vision, equilibrium, which is also your balance, and hearing. The sense organs involved are structurally more complex than those of general senses, but the same basic principles of the receptor function apply. The sense of smell, also called olfaction, is made possible by paired olfactory organs. These organs are located in the nasal cavity on either side of the nasal septum. The olfactory organs are made up of 2 layers, the olfactory equilibrium epithelium and the lamina propria. The olfactory equilibrium contains the olfactory receptor cells, supporting cells, and regenerative basal cells, or stem cells. This epithelium covers the inferior surface of the cribriform plate, the superior portion of the perpendicular plate, and the superior nasal conchae of the ethmoid. The second layer, the underlying lamina propria, consists of areolar tissue, numerous blood vessels, and nerves. This layer also contains olfactory glands. Their secretions absorb water and form thick pigmented mucus. What happens when you inhale through your nose? The air swirls within your nasal cavity. This turbulence brings airborne compounds to your olfactory organs. A normal, relaxed inhalation carries a small sample, about 2% of the inhaled air, to the olfactory organs. If you sniff repeatedly, you increase the flow of air across the olfactory epithelium, increasing the stimulation of the olfactory receptors. However, only the molecules of water-soluble and lipid-soluble materials can diffuse through the overlying mucus and stimulate those receptors. Olfactory receptor cells are highly modified neurons. The exposed tip of each receptor cell forms a prominent knob that projects beyond the epithelial surface. The knob is a base for up to 20 cilia-shaped dendrites that extend into the surrounding mucus. These dendrites lie parallel to the epithelial surface, exposing their considerable surface area to dissolved compounds called odorants. Olfactory reception begins with the binding of an odorant to a G protein-coupled receptor in the plasma membrane of an olfactory receptor cell dendrite. Between 10 and 20 million olfactory receptors fill an area of roughly 5 centimeters. If we take into account the exposed dendritic surface, the actual sensory area probably approaches that of the entire body surface. Yet, our olfactory sensitivities cannot compare with those of other vertebrates such as dogs, cats, and fish. A German Shepherd dog sniffing for smuggled drugs or explosives has an olfactory receptor surface 72 times greater than that of the nearby customs inspector. So the olfactory system is very sensitive. We know that. However, the activation of an afferent fiber does not guarantee an awareness of the stimulus. Considerable convergence takes place along the olfactory pathway, and inhibition of the innervating synapse can prevent the sensations from reaching the olfactory cortex of the cerebral hemispheres. The olfactory receptors themselves adapt very little to an outgoing stimulus. Rather, the central adaptation ensures that you quickly lose awareness of a new smell, but remain sensitive to others. Axons leaving the olfactory epithelium collect up to 20 or more bundles that penetrate the cribriform plate of the ethmoid bone to reach the olfactory bulbs of the cerebrum, where the first synapse occurs. Efferent fibers from the nuclei elsewhere in the brain also innervate neurons of the olfactory bulbs. This arrangement provides a mechanism for central adaptation or facilitation of olfactory sensitivity. Axons leaving the olfactory bulb travel along the olfactory tract to the olfactory cortex of the hypothalamus and portions of the limbic system. Olfactory stimulation is the only type of sensory information that reaches the cerebral cortex directly. All other sensations are relayed from processing centers in the thalamus. Certain smells can trigger profound emotional and behavioral responses, as well as the memories, due to parallel distribution of olfactory information to the limbic system and the hypothalamus. The perfume industry understands the practical implications of these connections and works to develop odors that will trigger sexual responses. So the human olfactory system can make subtle distinctions among 2,000 to 4,000 chemical stimuli. No apparent structural differences exist among the olfactory receptors, but the epithelium as a whole contains receptor populations with distinct sensitivities. At least 50 primary smells are known. It is almost impossible to describe these sensory impressions effectively. It appears like the CNS interprets each smell on the basis of the overall pattern of the receptor activity. The human olfactory organs can discriminate among many smells, but acuity varies widely, depending on the nature of the odorant. We detect many odorants in amazingly small concentrations. One example is mercaptan, an odorant commonly added to natural gas, propane, and butane, which are otherwise odorless. Because we can smell mercaptan in extremely low concentrations, about a few parts per billion, its addition enables us to detect gas leaks almost at once and take steps to prevent an explosion. The olfactory receptor population undergoes considerable turnover. Basal cells in the epithelium divide and differentiate to produce new receptor cells. This turnover is one of the few examples of neuronal replacement in adult humans. Despite this process, the total number of receptors declines with age, and those that remain become less sensitive. As a result, elderly people have difficulty detecting odors in low concentrations. This decline in the number of receptors accounts for grandma's tendency to use too much perfume and explains why grandpa's aftershave seems a little strong. They must apply more to be able to smell it themselves. Gustation, or taste, provides information about the foods and liquids that we eat and drink. The taste receptors, or gustatory receptors, are distributed over the superior surface of the tongue in adjacent proportions of the pharynx and the larynx. The most important taste receptors are on the tongue. By the time we reach adulthood, the taste receptors on the pharynx and larynx and epiglottis have decreased in number. Taste receptors in specialized epithelial cells form sensory structures called taste buds. An adult has about 10,000 taste buds. The surface of the tongue has numerous variously shaped epithelial projections called lingual papillae. The human tongue has 4 types of lingual papillae: the filiform papillae, the fungiform papillae, the velat papillae, and the folate papillae. The distribution of these lingual papillae varies by region. Filiform papillae provide friction that helps the tongue move objects around in the mouth, but do not contain taste buds. Each small fungiform papillae contains about 5 taste buds. There are up to 12 velat papillae that form a V near the posterior margin of the tongue, and each large velat papillae contains as many as 100 taste buds. The folate papillae are folds found on the lateral margins of the posterior tongue. The taste buds are recessed in the surrounding epithelium, isolated from the unprocessed contents of the mouth. Each taste bud contains about 40 to 100 receptor cells and many small stem cells called basal cells. The basal cells continually divide to produce daughter cells that mature in stages. The cells of the last stage are called gustatory receptor cells. Cells at all 3 stages, the basal, the transitional, and mature, are innervated by sensory neurons. Each gustatory receptor cell extends microvilli, sometimes called taste hairs, in its surrounding fluids through the taste pore, which is a narrow opening. Despite this relatively protected position, it's still a hard life. A typical gustatory receptor cell survives for only about 10 days before it's replaced. The taste buds are innervated by cranial nerves 7, 9, and 10. The facial nerve innervates all of the taste buds located on the anterior two-thirds of the tongue, from the tip to the line of the palate papillae. The glossopharyngeal nerve, or the ninth cranial nerve, innervates the palate papillae and the posterior one-third of the tongue. And the vagus nerve, or nerve 10, innervates the taste buds scattered on the surface of the epiglottis. The sensory afferent fibers carried by these cranial nerves synapse in the solitary nucleus of the medulla oblongata. The axons of the postsynaptic neurons then enter the medial lemniscus. There, the neurons join the axons that carry somatic sensory information on touch, pressure, and proprioception. After another synapse in the thalamus, the information is projected to the appropriate portions of the gustatory cortex in the insula. You have a conscious perception of taste as the brain correlates information received from the taste buds with other sensory data. Information about the texture of food, along with taste-related sensations such as peppery or burning hot, comes from the sensory afferents in the trigeminal cranial nerve. In addition, the level of stimulation from the olfactory receptors plays an overwhelming role in taste perception. The combination of taste and smell is what provides the flavor or the distinct quality of particular food or drink. You are several times more sensitive to taste when your olfactory organs are fully functional. By contrast, when you have a cold and your nose is stuffed up, airborne molecules cannot reach your olfactory receptors, so meals taste dull and unappealing. The reduction in flavor takes place even though the taste buds may be responding normally. Without accompanying olfactory sensations, you are now limited in the basic taste sensations provided by your gustatory receptors. You're probably already familiar with the four primary taste sensations: sweet, salty, sour, and bitter. There's some evidence for differences in sensitivity to taste along the axis of the tongue, with greater sensitivity to salty, sweet anteriorly and sour, bitter posteriorly. However, there are no differences in the structure of the taste buds. Taste buds in all portions of the tongue provide all four primary taste sensations. Humans have two additional taste sensations that are less widely known. Umami, which is derived from the Japanese word meaning deliciousness, is a pleasant savory taste corresponding to the flavor of beef broth, chicken broth, and Parmesan cheese. Umami is due to the presence of free glutamates in food. The distribution of these receptors is not known in detail, but they are present in the taste buds of the palate papillae. And water. A lot of people say that water has no flavor. However, research on humans and other vertebrates has demonstrated the presence of water receptors, especially in the pharynx. The sensory output of these receptors is processed in the hypothalamus and affects several systems that affect water balance and regulation of blood volume. For example, minor reductions in the secretion of antidiuretic hormone, or ADH, which is a hormone that regulates urination, occurs each time you take a long drink. The threshold for receptor stimulation varies for each of the primary taste sensations. Also, the taste receptors respond more readily to unpleasant than pleasant stimuli. For example, we are almost 1,000 times more sensitive to acids, which taste sour, than to either sweet or salty chemicals. We are 100 times more sensitive to bitter compounds than to acids. This sensitivity has a survival value because acids can damage the mucosal membranes of the mouth and the pharynx, and many of the potent biological toxins have an extremely bitter taste. Taste sensitivity differs significantly among individuals, and many conditions related to taste sensitivity are inherited. The best known example involves sensitivity to the compound PTC, or phenethylcarbamide, and this substance tastes bitter to some people, but is tasteless to others. PTC is not found in food, but compounds related to PTC are found in Brussels sprouts, broccoli, cabbage, and cauliflower. Our tasting abilities change with age. Many elderly people find that their food tastes bland and unappetizing, while children tend to find the same foods too spicy. And what accounts for these differences? Well, we begin life with more than 10,000 taste buds, but the number begins declining dramatically by the age of 50. The sensory loss becomes especially significant because, as we've already noted, older individuals also experience a decline in the number of olfactory receptors. So we rely more on vision than any other special sense. Our eyes contain our visual receptors. The eyes are elaborate structures that enable us not only to detect light, but to also create detailed visual images. And we'll begin our discussion of these organs with the accessory structures of the eye, which provide protection, lubrication, and support. So the accessory structures of the eye include the eyelids and the superficial epithelium of the eye, as well as the structures involved with production, secretion, and removal of tears. Eyelids and superficial epithelium of the eye. So the eyelids, or the palpebrae, are a continuation of the skin. Their continual blinking keeps the surface of the eye lubricated. They also act like windshield wipers, removing dust and debris. The eyelids can close firmly to protect the delicate surface of the eye. The palpebral fissures is the gap that separates the free margins of the upper and lower eyelids. The two eyelids are connected. However, at the medial canthus and the lateral canthus, the eyelashes along the margins of the eyelids are very robust hairs. They help to prevent foreign matter, including insects, from reaching the surface of the eye. The eyelashes are associated with an unusually large sebaceous gland. Along the inner margin of the lid are small modified sebaceous glands called tarsal glands or meibomian glands. They secrete lipid-rich product that helps to keep the eyelids from sticking together. At the medial canthus, the lacrimal chronicle, which is a mass of soft tissue, contains glands producing the thick secretions that contribute to the gritty deposits that sometimes appear after a good night's sleep. Bacteria occasionally invade and infect these various glands. A chalazion is a cyst that results from an infection of a tarsal gland. An infection in a sebaceous gland, also associated with an eyelash, produces a painful localized swelling known as a stye. The skin covering the visible surface of the eyelid is very thin. Deep to the skin lie the muscle fibers of the orbicularis oculi and the levator palpebrae superius muscles. These skeletal muscles close the eyelid and raise the upper eyelid. The epithelium covering the inner surfaces of the eyelids and the outer surface of the eyeball is called the conjunctiva. It is a mucosous membrane covered by a specialized stratified squamous epithelium. The palpebrae conjunctiva covers the inner surface of the eyelids. The ocular conjunctiva, or bulbar conjunctiva, covers the anterior surface of the eye. The ocular conjunctiva extends to the edges of the cornea, a transparent part of the outer fibrous layer of the eye. The cornea is covered by a very delicate squamous corneal epithelium, 5 to 7 cells thick, that is continuous with the ocular conjunctiva. A constant supply of fluid washes over the surface of the eyeball, keeping the ocular conjunctiva and the cornea moist and clean. Mucus cells in the epithelium assist the accessory glands in lubricating the surfaces of the conjunctiva to prevent drying out and friction. Conjunctivitis, or pink eye, is an inflammation of the conjunctiva. The most obvious sign, which is redness, is due to the dilation of blood vessels deep to the conjunctival epithelium. This condition may be caused by pathogenic infection or by physical, allergic, or chemical irritation of the conjunctival surface. A constant flow of tears keeps the conjunctival surfaces moist and clean. Tears reduce friction, remove debris, prevent bacterial infection, and provide nutrients and oxygen to portions of the conjunctival epithelium. The lacrimal apparatus produces, distributes, and removes tears. The lacrimal apparatus of each eye consists of 4 parts: a lacrimal gland with associated ducts, paired lacrimal caniculi, a lacrimal sac, and a nasolacrimal duct. The fornix of the eye is a pocket created where the palpebral conjunctiva becomes continuous with the ocular conjunctiva. The lateral portion of the superior fornix receives 10 to 12 ducts from the lacrimal gland or tear gland. This gland is about the size and shape of an almond, measuring roughly 12 to 20 millimeters. It nestles within a depression of the frontal bone just inside the orbit and superior and lateral to the eyeball. The lacrimal gland normally provides the key ingredients and most of the volume of the tear that bathe the conjunctival surfaces. The lacrimal secretions supply nutrients and oxygen to the corneal cells by diffusion. The lacrimal secretions are watery and slightly alkaline, and they contain the antibacterial enzyme lysosome and the antibodies that attack pathogens before they enter the body. The lacrimal gland produces about 1 milliliter of tears a day. And once the lacrimal secretions have reached the ocular surface, they mix with the products of the accessory gland and the oil secretions of the tarsal gland. The result is a superficial oil slick that aids lubrication and slows evaporation. At the medial canthus in an area known as the lacrimal lake or lake of tears, the lacrimal lake covers the lacrimal crew-knuckle, which bulges anteriorly. The lacrimal puncta, which is 2 small pores, drain the lacrimal lake. They empty into the lacrimal canicula, small canals that turn and lead to the lacrimal sac, which nestles within the lacrimal sulcus of the orbit. From the inferior portion of the lacrimal sac, the nasolacrimal duct passes through the nasolacrimal canal, formed by the lacrimal bone of the maxilla. This duct delivers tears to the nasal cavity on that side. The duct empties into the inferior meatus, or a nasal passageway inferior to the lateral and inferior to the nasal concha. And when a person cries, tears rushing into the nasal cavity produce a runny nose. If the lacrimal puncta can't provide enough drainage, the lacrimal lake overflows and tears stream across the face. Your eyes are extremely sophisticated and visual instruments. They are more versatile and adaptable than the most expensive cameras, and yet compact and durable. Each eye is a slightly irregular spheroid, or a little smaller than a ping pong ball. An eye has an average diameter of about 24 millimeters and a weight of about 8 grams. Within the orbit, the eyeball shares space within the extrinsic eye muscles, the lacrimal gland, and the cranial nerves and blood vessels that supply the eye and the adjacent portions of the orbit and face. Orbital fat cushions and insulates the eye. The wall of the eye has 3 distinct layers, formerly called the tunics. The first, an outer fibrous layer. The second, an intermediate vascular layer. And third, the deep inner layer. The visual receptors or photoreceptors are located in the inner layer. The eyeball itself is hollow and filled with fluid. Its interior can be divided into 2 cavities, the anterior and the posterior. The anterior cavity has 2 chambers. The anterior chamber, which is between the cornea and the colored portion known as the iris, and the posterior chamber, which is between the iris and the lens. A clear watery fluid called aqueous humor fills the entire anterior cavity. The posterior cavity, or vitreous chamber, contains a gelatinous substance called the vitreous body. The fluid part of this body is vitreous humor. The vitreous body and the aqueous humor help to stabilize the shape of the eye. The fibrous layer, or the outermost layer of the eye, consists of the sclera and the cornea. The fibrous layer, 1, supports and protects, 2, serves as an attachment site for the extrinsic eye muscles, and 3, contains structures that assist in focusing. The sclera, or the white of the eye, covers most of the ocular surface. The sclera consists of dense fibrous connective tissue containing both collagen and elastic fibers. This layer is the thickest over the posterior surface of the eye near the exit of the optic nerve. The sclera is the thinnest over the anterior surface. The 6 extrinsic eye muscles insert on the sclera, bending their collagen fibers with those of the fibrous layer. The surface of the sclera contains small blood vessels and nerves that penetrate the sclera to reach the internal structures. The network of small vessels interior to the ocular conjunctiva generally does not carry enough blood to lend an obvious color to the sclera. On close inspection, however, the vessels are visible as red lines against a white background of collagen fibers. The transparent cornea is a structure continuous with the sclera. The border between the two is called the corneal limbus. Deep to the delicate corneal epithelium, the cornea consists primarily of a dense matrix containing multiple layers of collagen fiber that are organized as not to interfere with the passage of light. The cornea has no blood vessels. Its superficial epithelial cells contain oxygen and nutrients by diffusion from the tears that flow across the free surface. The cornea also has numerous free nerve endings, making it the most sensitive portion of the eye. Damage to the cornea may cause blindness, even though the functional components of the eye, including the photoreceptors, are perfectly normal. The cornea has a very restricted ability to repair itself, and for this reason, corneal injuries must be treated immediately to prevent serious vision losses. Restoring vision after corneal scarring generally requires replacing the cornea through a corneal transplant. Corneal replacement is probably the most common form of transplant surgery. Such transplants can be performed between unrelated individuals because there are no blood vessels to carry white blood cells, which attack foreign tissue into the area. Corneal grafts are obtained from donor eyes. For best results, the tissues must be removed within 5 hours after the donor's death. So the vascular layer, or the uvea, is a pigmented region that includes the iris, the ciliary body, and the choroid. It contains numerous blood vessels, lymphatic vessels, and intrinsic muscles of the eye. This middle layer, 1, provides a route for blood vessels and lymphatics that supply the tissue of the eye. 2, regulates the amount of light that enters the eye. 3, secretes and reabsorbs the aqueous humor that circulates within the chambers of the eye. And 4, controls the shape of the lens, which is an essential part for focusing process. The iris is visible through the transparent corneal surface, and the iris contains blood vessels, pigmented cells, and 2 layers of smooth muscle fibers called pupillary muscles. When these cells contract, they change the diameter of the pupil, the central opening of the iris. There are 2 types of pupillary muscles, dilators and constrictors. The autonomic nervous system controls both muscle groups. For example, parasympathetic activation in response to bright light causes the pupils to constrict. Sympathetic activation in response to dim light causes the pupils to dilate. The body of the iris consists of highly vascular, pigmented, loose connective tissue. The anterior surface has no epithelial covering. Instead, it has an incomplete layer of fibroblasts and melanocytes. Melanocytes are also scattered within the body of the iris. The posterior surface is covered by a pigmented epithelium that is part of the inner layer and contains melanin granules. So how is your eye color determined? Our genes influence the density and distribution of melanocytes on the anterior surface and interior of the iris, as well as the density of the pigmented epithelium. When connective tissues of the iris contain few melanocytes, light passes through and bounces off the pigmented epithelium. The eye then appears blue. But individuals with green, brown, or black eyes have an increasing number of melanocytes in the body and on the surface of the iris. The eyes of people with albinism, whose cells do not produce melanin, appear a very pale gray or bluish gray. At its periphery, the iris attaches to the anterior portion of the ciliary body, a thickened region that begins deep to the junction between the cornea and the sclera. The ciliary body extends posteriorly to the level of the ora serrata, the serrated anterior edge of the thick neural part of the inner layer or retina. The bulk of the ciliary body consists of the ciliary muscle, a ring of smooth muscle that projects into the interior of the eye. The epithelium covering this muscle has numerous folds called ciliary processes. The ciliary zonule is the ring of fibers that attaches to the lens of the ciliary processes. The connective tissue fibers hold the lens in place posterior to the iris and centered on the pupil. As a result, any light passing through the pupil also passes through the lenses. The choroid is a vascular layer that separates the fibrous layer and the inner layer posterior to the ora serrata. The choroid is covered by the sclera and attached to the outermost layer of the retina. An extensive capillary network in the choroid delivers oxygen and nutrients to the retina. The choroid also contains melanocytes, which are especially numerous near the sclera. The inner layer, or the retina, is the innermost layer of the eye. It consists of a thin outer layer called the pigmented part and a thick inner layer called the neural part. The pigmented part of the retina absorbs light that passes through the neural part, preventing light from bouncing back through the neural part and producing visual echoes. The pigmented cells also have important biochemical interactions with the retina's light receptors, which are located in the neural part of the retina. In addition to light receptors, the neural part of the retina contains supporting cells and neurons that do preliminary processing and integration of visual information. The 2 layers of the retina are normally very close together, but not tightly interconnected. The pigmented part of the retina continues over the ciliary body and the iris, but the neural part extends anteriorly only as far as the ora serrata. The neural part of the retina forms a cup that establishes the posterior and lateral boundaries of the posterior cavity. Organization of the retina. In sectional view, the neural part of the retina contains several layers of cells. The outermost layer, closest to the pigmented part of the retina, contains the photoreceptors, which are the cells that detect light. The eye has two main photoreceptors, rods and cones. Rods do not discriminate among colors of light. Highly sensitive to light, they enable us to see in dimly lit rooms, as twilight and in pale moonlight. Cones give us color vision. We have three types of cones, and each type consists of different visual pigment, red, green, and blue. A person who sees three primary colors has normal color vision, and thus is referred to as a trichromat. Their stimulation in various combinations enables us to perceive different colors. Cones give us sharper, clearer images than rods, but cones require more intense light. If you sit outside at sunset with your textbook and open to a colorful illustration, you can detect the gradual shift in your visual system from a cone-based vision, which will give you a clear image in full color, to rod-based vision, which is relatively grainy image in black and white. A third type of photoreceptor is the intrinsically photosensitive retinal ganglion cell, or the IPRGC. The photopigment in the IPRGC is melanopsin. These cells are known to respond to different levels of brightness and influences the body's 24-hour circadian rhythm. Rods and cones are not evenly distributed across the retina. Approximately 125 million rods form a broad band along the periphery of the retina. As you move away from the periphery towards the center of the retina, the density of the rods gradually decreases. In contrast, most of the roughly 6 million cones are concentrated in the area where visual image arrives after it passes through the cornea and the lens. This region, known as the macula, has no rods. The very highest concentration of cones occurs at the center of the macula, an area called the fovea centralis. It is usually simply referred to as the fovea. The fovea is the site of the sharpest vision. When you look directly at an object, its image falls in the portion of the retina. An imaginary line drawn from the center of that object through the center of the lens to the fovea establishes the visual axis of the eye. So what are some of the visual consequences of the distribution of photoreceptors? When you look directly at an object, you are placing its image on the fovea, the center of color vision. You see a very good image as long as there is enough light to stimulate the cones, but in very dim light, cones cannot function. That's why you can't see a dim star if you stare directly at it. But you can see it if you shift your gaze to one side or the other. Shifting your gaze moves the image of the star from the fovea, where it does not provide enough light to stimulate the cones, to the periphery, where it can affect more sensitive rods. Rods and cones synapse with about 6 million neurons called bipolar cells. These cells turn in synapse within the layers of the neurons called ganglion cells, which lie adjacent to the posterior cavity. A network of horizontal cells extends across the neural part of the retina at the level of the synapse between the photoreceptors and the bipolar cells. A comparable layer of amacrine cells occurs where the bipolar cells synapse with ganglion cells. Horizontal and acrimin cells can facilitate or inhibit communication between photoreceptors and ganglion cells, altering the sensitivity of the retina. And these play an important role in the eye's adjustment to dim or brightly lit environments. So axons from an estimated 1 million ganglion cells converge on the optic disc, which is a circular region just medial to the fovea. The optic disc is the origin of the optic nerve. From this point, the axons turn, penetrate the wall of the eye, and proceed towards the diencephalon. The central retinal artery and the central retinal vein, which supply the retina, pass to the center of the optic nerve and emerge on the surface of the optic disc. The optic disc has no photoreceptors or other structures typical of the rest of the retina. The optic disc is commonly called the blind spot because light striking this area goes unnoticed. So why don't you see a blank spot in your field of vision? The reason is that involuntary eye movements keep the visual image moving and allow your brain to fill the missing information. However, There are some activities you can do which can prove that the blind spot actually exists in your field of vision if you're interested in them. So recall that the ciliary body and the lens divide the interior of the eye into large posterior cavity and the smaller anterior cavity made up of anterior and posterior chambers. Both those chambers are filled with the fluid aqueous humor. The posterior cavity also contains aqueous humor, but a gelatinous substance known as the vitreous body that takes up most of its volume. Aqueous humor is a fluid that circulates within the anterior cavity. It passes from the posterior chamber to the anterior chamber through the pupil. It also freely diffuses through the posterior cavity and across the surface of the retina. Its composition resembles that of cerebrospinal fluid. Aqueous humor forms through the active secretion by epithelial cells of the ciliary body's ciliary processes. The epithelial cells can regulate its composition. Because aqueous humor circulates, it provides an important route for nutrients and waste transport. It also serves as a fluid cushion. The eye is filled with fluid, and fluid pressure provided by the aqueous humor helps retain the eye's shape. Fluid pressure also stabilizes the position of the retina, pressing the neural part against the pigmented part. In effect, the aqueous humor acts like the air inside of a balloon. The eye's intraocular pressure can be measured in the anterior chamber, where the fluid pushes against the inner surface of the cornea. Intraocular pressure is most often checked by applanation tonometry. This procedure involves placing a small flat disc on the anesthetized cornea to measure the tension. Normal intraocular pressure ranges from about 12 to 21 aqueous humor is secreted into the posterior chamber at a rate of 1 to 2 milliliters per minute, and it leaves the anterior chamber at the same rate. After filtering through a network of connective tissues located near the base of the iris, aqueous humor enters the scleral venous sinus, a passageway that extends completely around the eye at the level of the corneal limbus. Collecting channels deliver the aqueous humor from this channel to the veins of the sclera. Aqueous humor is removed and recycled within a few hours of its formation. The rate of removal normally keeps pace with the rate of generation of ciliary processes. The posterior cavity of the eye contains the vitreous body, which is a gelatinous mass. Its fluid portion, called the vitreous humor. The vitreous body helps to stabilize the shape of the eye. Otherwise, the eye might distort as the extrinsic eye muscles change its position within the orbit. Specialized cells embedded in the vitreous body produce the collagen fibers and photoglycans that account for the gelatinous consistency of the mass. Unlike the aqueous humor, the vitreous body is formed during the development of the eye and is not replaced. The lens lies posterior to the cornea and is held in place by the ciliary zonule that originates in the ciliary body of the choroid. The primary function of the lens is to focus the visual image on the photoreceptors. The lens does so by changing its shape. The lens consists of concentric layers of cells that are precisely organized. A dense fibrous capsule covers the entire lens. Many of the capsular fibers are elastic. Unless an outside force is applied, they will contract and make the lens spherical. Around the edges of the lens, these capsular fibers intermingle with those of the ciliary zonule. The cells in the interior of the eye are called lens fibers. These highly specialized cells have lost their nuclei and other organelles. They are long and slender and are filled with transparent proteins called crystallins. These proteins give the lens both its clarity and its focusing power. Crystallins are extremely stable proteins. They remain intact and functional for a lifetime. The transparency of the lens depends on the crystalline proteins maintaining a precise combination of structural and biochemical characteristics. Because these proteins are not renewed, any modifications that they undergo over time can accumulate and result in the lens losing its transparency. This abnormality is known as a cataract. Cataracts can result from injuries, radiation, or reaction to drugs. Senile cataracts, however, are a natural consequence of aging and are the most common form. Over time, the lens turns yellowish and eventually begins to lose transparency. As the lens becomes cloudy, the individual needs brighter and brighter light for reading. Visual clarity begins to fade. If the lens becomes completely opaque, the person will be functionally blind, even though the photoreceptor is normal. Cataract surgery involves removing the lens, either intact or after it has been shattered with high-frequency sound waves. The missing lens is then replaced by an artificial substitute. Vision is fine-tuned with glasses or contact lenses. So the retina has about 130 million photoreceptors. Each one monitors the light striking a specific site on the retina. The processing of information from all the receptors produces the visual image. The eye is often compared to a camera. To provide useful information, the lens of the eye, like a camera lens, must focus on the arriving image. To say that an image is in focus means that the rays of the light arriving from the object strike the sensitive surface of the retina, or the semiconductor device that records light electrically into the digital camera, in precise order, so as to form a miniature image of the object. If the rays are not perfectly focused, the image is blurry. Focusing normally occurs in two steps, as the light passes first through the cornea and then the lens. Light is refracted or bent when it passes from one medium to another medium with different density. You can see this effect if you stick a pencil into a glass of water. Notice that the shaft of the pencil appears to bend sharply at the air-water interface. This effect occurs when light is refracted as it passes into the air from the much denser water. In the human eye, the greatest amount of refraction occurs when the light passes from the air into the corneal tissues, which have a density close to that of water. When you open your eyes underwater, you cannot see clearly because refraction at the corneal surface has been largely eliminated. Light passes unbent from one watery medium to another. Additional refraction takes place when the light passes from the aqueous humor into the relatively dense lens. The lens provides the extra refraction needed to focus the light rays from an object toward the focal point, a specific point of intersection on the retina. The distance between the center of the lens and its focal point is the focal distance of the lens. Whether in the eye or a camera, the focal distance is determined by two factors. One, the distance of the object from the lens. So the closer an object is to the lens, the greater the focal distance. And two, the shape of the lens. The rounder the lens, the more refraction occurs. So a very round lens has a shorter focal distance than a flatter one. Accommodation is the automatic adjustment of the eye to give us clear vision. During accommodation, the lens becomes rounder to focus on the image of a nearby object on the retina. The lens becomes flatter to focus the image of a distant object on the retina. So how does the lens change shape? The lens is held in place by the ciliary zonule that originates in the ciliary body. Smooth muscle fibers in the ciliary body act like sphincter muscles. When the ciliary muscle contracts, the ciliary body moves towards the lens, thereby reducing the tension in the ciliary zonule. The elastic capsule then pulls the lens into a rounder shape and increases the refractive power of the lens. This enables it to bring light from nearby objects into focus on the retina. When the ciliary muscles relaxes, the ciliary zonule pulls at the circumference of the lens, making the lens flatter. The greatest amount of refraction is required to view objects that are very close to the lens. The inner limit of clear vision, known as the near point of vision, is determined by the degree of elasticity in the lens. Children can usually focus on something about 7 to 9 centimeters from the eye, but over time the lens becomes stiffer and less responsive. A young adult can usually focus on objects about 10 to 20 centimeters away, and as we age, this distance gradually increases. The near point at age 60 is about 83 centimeters. If light passing through the cornea and lens is not refracted properly, the visual image will be distorted. In the condition called an astigmatism, the degree of the curvature of the cornea or the lens varies from one axis to another. Minor astigmatism is very common. The image distortion may also be minimal, that people are unaware of the condition. So we've been discussing light that originates at a single point, either near or far from the viewer. An object that we see as a really complex light source must be treated as a number of individual points. Light from each point is focused on the retina, as in the figure that you see over here to the right. The result is a miniature image of the original, but the image arrives upside down and reversed from left to right. So why does that image arrive upside down? Again, consider the figure to the right, a sagittal section through the eye that is looking at a telephone pole. The image at the top of the pole lands at the bottom of the retina, and the image at the bottom hits the top of the retina. Now consider the figure below that, a horizontal section through the eye that's looking at a picket fence. The image of the left edge of the fence falls on the right side of the retina, and the image of the right side falls on the left side of the retina. The brain compensates for this image reversal so that you are not aware of any difference between the orientation of the image on the retina and that of the object. As a result, the crossover of light rays is shown in the lens, but it actually occurs very close to the fovea. So how well you see, or your visual acuity, is rated by comparison to a normal standard. The standard vision rating of 20/20 is defined as the level of detail seen at distance from 20 feet by a person with normal vision. That is, a person with a visual acuity of 20/20 sees clearly at 20 feet what should normally be seen at 20 feet. Vision rated as 20/15 is better than average because at 20 feet, the person is able to see details that would be clear to a normal eye only at the distance of 15 feet. Conversely, a person with 20/30 vision must be 20 feet from an object to discern details that a person with normal vision could make out at a distance of 30 feet. When visual acuity falls below 2200, even with the help of glasses or contact lenses, the individual is considered to be legally blind. There are probably fewer than 400,000 legally blind people in the United States, and more than half are over 65 years old. The term blindness implies a total absence of vision due to the damage to the eyes or the optic pathways. Common causes of blindness include diabetes mellitus, cataracts, glaucoma, corneal scarring, retinal detachment, accidental injuries, and hereditary factors that are still not really understood. An abnormal blind spot, or scotoma, may appear in the field of vision at positions other than the optic disc. A scotoma is permanent and may result from the compression of the optic nerve, damage to the photoreceptors, or central damage along the visual pathway. Floaters are small spots that drift across the field of vision, and they are generally temporary and result from blood cells or cellular debris in the vitreous body. You may have seen floaters if you've ever stared at a blank wall or a white sheet of paper and noticed little spots. So with this picture is where we're going to stop off for the first part of this PowerPoint. In the next PowerPoint, or part 2 of 9.3, we'll cover visual physiology, equilibrium, and hearing.